of speciation, I'd like to talk about a third type of speciation, sympatric speciation. And this is different from allopatric speciation, which relies upon geographic isolation to allow species to differentiate, and parapatric speciation, which relies upon differential selection across environmental gradient to result in speciation. Sympatric speciation is by far one of the rarest and most controversial forms of speciation because it is speciation that occurs without barriers and without gradients, and you have a new species appearing within the range of an existing species in the face of great potential for gene flow. So we talked about how gene flow is a great homogenizer of genetic variation between populations. That's why allopatric speciation is so easy relatively, because the barrier prevents that gene flow from occurring. In the case of the parapatric speciation, that gene flow is reduced by selection against hybrids along that environmental gradient. But what about our sympatric speciation? Well, we have a new species forming in the face of potential gene flow, and gene flow acting as a homogenizer. So the biggest theoretical objection to sympatric speciation is the fact that there is great potential for gene flow, and gene flow would tend to wash out or dilute any of the uniqueness that you would associate with being part of a new species. Now, there is one model which is commonly described as an example of sympatric speciation, and that's the case of the apple maggot fly, Rhagoletis pulmonella. And this is a pest species of cultivated apples. In North America, apples were introduced in the 1600s, and starting with the 1800s, there was the first report of them being parasitized by this particular fly. And they hadn't been seen on apples before that time, and the question was, well, what is this pest and where did it come from? Now, the life history of Rhagoletis pulmonella is where we should probably start off. It is a fly that spends its life in close association with its host fruit. That is, it seeks out fruit, it mates and courts on the surface of the fruit, it lays its eggs inside the fruit. When the fruit fall off the tree, those eggs develop in them. For a certain bit of time up until the fall. Then there is a period of diapause that happens in the fall and allows them to go into essentially a dormant metabolic state. So they overwinter, and then in the spring, in the summer, the adults emerge from their pupae, and they start the cycle all over again. Now, it was noticed that the apple maggot flies looked an awful lot very similar to the hawthorn maggot flies. And hawthorns are an endemic, a native North American species that is found in North America. That there's big differences between hawthorns and apples. Obviously, hawthorns are much smaller, than apples. They're much larger. And it was suggested even in the 1800s that this was a case of a new species arising because of switching in hosts. So this was a new host form, and that this change in ecological usage was central to this formation of this new species. But if we're going to think about host switching, we need to think about what would be the pros and cons associated with host switching. Well, one pro would be obviously while you're moving from a hawthorn to an apple, there's an awful lot more things to consume. Apples are much bigger, but apples are actually relatively less nutritious from the perspective of these maggots than hawthorns. So there's much lower survival of the maggots on apples than on hawthorns. It could be that their size is advantageous, so that because they're larger, that the eggs that are laid can be laid deeper in them, and thus avoid the parasitic wasps that also lay their eggs that feed on their larvae. So there could have been a good incentive for individuals to start laying the eggs in other species, and a selective advantage to become specialized as exploiting that particular host, the apples, and form this new species. There could be a good selection pushing that. Now, is this a case of sympatric speciation? Well, let's look at some of the evidence. So from a sympatric, the populations are sympatric, that is, they're found at the same location. Sym meaning the same, patric meaning country, so same country. So hawthorns and apples are found in the same places geographically, so there's no barriers, there's no clear gradients between them. Morphologically, they are identical. You cannot see any differences on the surface of them. But if you do look at them genetically, they have very distinct genetic profiles. They have different frequencies of specific genes that are strongly associated with the length and depth of the diapause, that overwintering. So from a morphological perspective, they're indistinguishable. Genetically, they're quite easy to tell apart. Now, in a series of experiments, it's found that there's strong ecological preference for their own host plants. That is, given a choice, they will go and lay eggs on their original host species, host brood. But there is small levels of hybridization between the host forms, 4 to 6%. But when it does happen, they are viable and fertile. So we see big ecological differences, but technically there are no reproductive barriers. So there's a great potential for gene flow. So really interesting situation there. And what seems to be the main driving force for this evolution that's reducing the amount of potential gene flow is that each one of these different host forms is strongly tied into the phenology, that is the timing of their hosts. So in the case of hawthorns, which mature in October, you see the hawthorn types eclosing from their pupae a bit earlier in September to capitalize on the fact that they mature, and then they mate and they have good hosts in which they can lay eggs. Apples, however, mature much sooner in the season. August and September is when you find apples, and so the apple host flies eclose even a little earlier, and so you can see there is a big ecological difference between them, that each one is being selected divergently to lay eggs on a different kind of host fruit, and that has different implications about when they mature. So any apple host forms that wait round, that delay mating and reproduction until October, are probably going to die off before then. They're waiting too long, and that most of the hawthorns are closing too late to even make use of that apple resource. So there's strong divergent disruptive selection acting on it. So that's the simple textbook example of what happens. But in fact, it's much more complicated. I'll jump over that question. I won't talk about it today. But let's think about some of the more complex issues, which aren't really covered as much, because it's really not that simple a story. Because the genes, those key 6 loci that are really important for understanding the differences in diapause length, the depth of their overwintering, are found in areas of chromosomal inversion. So as we've talked about in the first week of class, we have cases where whole chunks of the chromosome get cut out, switched around, and go back in different order. And about 50% of the Rhagoletis genome exhibits these inversion polymorphisms. So there are many different ways that chromosomes can be altered, and in the case of inversions, that broken segment is attached in reversed orientation. Now the consequences is that during the process of sexual reproduction, the production through meiosis of gametes, that those areas do not line up perfectly well and do not undergo genetic recombination. So there are 2 different structures of the chromosomes. There is a set of chromosomes that do really well for exploiting apples very well, and ones that exploit hawthorns really well, but the genes involved in diapause are found in that area of inversion, which means that if they end up in an offspring together, that they don't have recombination breaking them up. Recombination, remember, is when you have the arms of the chromosomes crossing over and breaking up combinations. So if gene flow between populations is a bad thing, for differentiation, recombination is a bad thing because essentially it's the mixing of genomes that you would have if you had that low levels of mating and producing viable fertile offspring. If there was recombination occurring, then it would mean that you would have the movement of hawthorn adapted genes into the apple maggot genome and vice versa. But we don't see that occurring because there is no, the cellular machinery that would normally allow that crossing over doesn't work when you have these two different polymorphic chromosomes coming together, which means that they can maintain their genomic uniqueness. It means that essentially that they're saved from this mixing. So it really helps facilitate that local adaptation because genes that benefit apples can accumulate, alleles that benefit apples can accumulate on one type of chromosome, and those that benefit hawthorns can accumulate another chromosome, and we don't get mixing of the two because they're in areas where there is no recombination. And then it's even more complicated because this is not something that has just happened overnight. Looking at the history of these flies, it looks like that there are different haplotypes, that is, different types of genetic profiles are found in different areas of North America. And the hawthorn maggot flies extend all the way from the northeast all the way down into Mexico, and that there are the type of inversion chromosomes that would benefit apples tend to be found initially in Mexico and Texas, and then have subsequently have moved their way up. And we think what happened was at some point there was a barrier, an allopatric barrier between them, and probably some populations on one side or the other, there was genetic drift occurring, and they evolved in different ways. That the north population evolved in one way, favoring one type of phenotype. The southern populations evolved in a different way, favoring a different kind of combination of loci. And then later they came into secondary contact, and that those particular alleles which are well adapted to that would end up being useful for colonizing and making that host switch for apples, moved up by genetic drift and through migration up into the north, where they were normally at a selective disadvantage when dealing with hawthorns because the timing of eclosion was off. But when they came across apples, when apples were introduced, they were well suited to exploiting that new environmental resource. So there was a good chance for them to occur. So it isn't a simple case of sympatric speciation. There probably was some degree of allopatric speciation to begin with. So very, very complicated. Now, the last topic I want to talk about is the idea of what does it take to be speciated? Is this a gradual change over time, or is it very fast? Does it involve few or many traits? We're going to build on this idea in the future, and we're going to talk about gradual change a great deal next week, but I want to talk about some rapid change. And we can see rapid changing occur when we have sudden, unexpected combinations of chromosomes. And in the first week of class, we talked about how that can occur either through failures of homologous pair separations in meiosis 1 or 2, in meiosis 1, or chromatid separation during meiosis 2, ending up with gametes that have more or less than expected number of chromosomes. But we can also involve entirely doubling or tripling or quadrupling, etc., of genomes. We call these polyploids. And polyploid individuals often have a really different genetic makeup, where they end up with more copies of different genes, and species can evolve, and new species can arise via these polyploid events. So there's two different kinds of these polyploid events. We call them autopolyploid, auto meaning self. So all the poly meaning many and ploid is copies. So there are many copies coming from the self. And in the case of the evening primrose, which has 14 chromosomes, we see that this is the parent species for this other species of primrose, in which there's been a doubling of their genome. So they have 28 chromosomes instead of 14 chromosomes. They are morphologically different. They are genetically incompatible, reproductively incompatible. And this is a case of a species that arises within the home range of the original species, suddenly within 1 or 2 generations. So an example of sympatric speciation, speciation occurring within the home range suddenly. And this is one way in which they can quickly overcome those gene flow, because by doubling chromosomes, they're no longer compatible. Now, you can actually make your own autoploids if you want by treating plants with colchicine. So it's actually used in many cases in horticulture to try and make fruits and leaves bigger. Interesting idea. We'll talk a bit more of that in the future. The other type of sympatric speciation that can arise for evolution of a brand new species all of a sudden is kind of allopolyploidy. And allo meaning different, the same way as we had allopatric, which was different countries. This is allopolyploidy, so different source of many genomes. In the case of the common hemp nettle, that is has 2 parents. It's a hybrid species that has resulted from the hybridization between the gametes of an Edmonton hemp nettle and a bee nettle, and it is now genetically distinct, morphologically distinct, and its own unique species. So again, a case of sympatric speciation that arises really quickly. Now, the big takeaway message we'll be taking away from this week is that how species are complex. We've seen that in species concepts, in our species concepts, and it's really hard for us to sort of track them down. So don't try to put species into a single box. They're really hard to define. Have a nice weekend, and I will see you next week.